Greetings, everyone. I'm Patricia Terrell with the American Physiological Society. I'm the Meetings and Program Coordinator and the moderator for today's webinar. Thank you for taking time out of your busy schedule to join us for this very exciting presentation titled Using Ossabaw Miniature Swine Models to Mimic Human Disease. And before we begin, here are a few important housekeeping notes. The webinar will be recorded and made available for on-demand viewing. And in order to maintain scientific integrity, we do ask that you refrain from taking pictures or videos in whole or in part of the presentation. Now, if you look at the bottom of your Zoom screen, you will find a Q&A and chat feature. And we ask that during the webinar that you submit your questions using the Q&A feature. That way we'll receive them promptly, and you can submit your questions at any time during the presentation. In addition, you will also find that live captions are available, and you can do so by clicking the live transcript icon, also located at the bottom of your Zoom screen. Now, before I introduce Dr. Sturek, please, I'd like for you to participate in a quick poll because we want to find out which animal models that you currently employ in your current workplace. And we will begin to launch that poll right now. Wow. Wow, these are amazing results, which I'm sure Dr. Sturek will be happy to see these results and would be very helpful in his presentation. So thank you so much for your feedback. And so what I'd like to do is go ahead and have Jake to stop sharing his screen. And what I'd like to do is start the process of introducing our distinguished speaker, Dr. Michael Sturek. There's so much to say about who he is and what he does, but he is the Chief Scientific Officer of CorVus Biomedical, LLC, and the CorVus Foundation. He is an adjunct professor of biomedical engineering at Purdue University and an adjunct professor of preclinical physiology and molecular imaging at Odense University Hospital and the University of Southern Denmark. He has 35 years of experience in academia, teaching, and conducting research on cardiometabolic disease. He was also a medical school department chair for 15 years. The general aim of his research program is to understand cellular and molecular calcium signaling mechanisms for in vivo comorbidities in cardiometabolic disease. And today, Dr. Sturek will present CorVus Biomedical's research on this innovative animal model, its many uses in exploring translational interventions, and the gene editing of Ossabaw miniature pigs. And it is indeed my honor and delight to bring forth Dr. Michael Sturek. Okay, Dr. Sturek, you can join us via video, and I'll make sure that you're all set.
You actually can't. It says you cannot start your video because the host has stopped it.
Well, we'll have to rectify that. Just one moment. Okay.
Okay. Yeah, there we go.
Great. So I can see you very well and I can hear you, and I will now have you to start your presentation.
All right, can we see that?
Not yet.
Not yet.
So share your screen.
Okay.
Okay.
Now we're good to go.
We are good to go. So I turn the baton over to you, Dr. Sturek.
Okay, thank you very much.
You're welcome.
and thank you very much, Patricia, for that very nice introduction. And so I'd like to thank everybody for joining today too. I'll get going right away here. And I'll start off with other thanks. And that is first, a big thanks to our CorVus team, Mohammed Alouche, Jim Bird. They formed the company with me, Rabia, as our CFO, we couldn't live without, and our technical staff, Josh, Jane, and Muhammad Ali Alush, and another Alush, who did an excellent job as an undergraduate fellow with us. And Daryl Ragland is our veterinarian, who's probably the best swine veterinarian in the country. And I'd also like to thank the American Physiological Society for letting us have this webinar. And Jake White and Patricia Terrell, you're super preparing me for this presentation. So this is an outline of what I want to go through today, from starting off with an overview and then touching on this list of different uses of the Ossabaw miniature swine. And what you're going to find is that it's not detailed enough for some. I give you kind of snippets on it, so you can, and if you want more detailed information, you can ask me after the presentation. There's just not enough time to tell you everything I would love to tell you. So as an overview here, obesity drives is one of the major drivers for metabolic syndrome or insulin resistance syndrome, and which can lead to type 2 diabetes and then a lot of subsequent comorbidities. The long-term goals of our CorVus research, both for the foundation and for the for-profit company, is to develop animal models for mimicking human disease, prediabetes, obesity, prediabetes, type 1 diabetes, and type 2 diabetes. So we're kind of greedy here. We want to try and do it all. And second, develop therapies for coronary heart disease, which is a major long-term complication in obesity and diabetes patients, and that's what we've been studying for quite some time. And thirdly, rapid translation of diabetes therapies to the clinic. And indeed, this translation to the clinic is it's really our theme song here, and that's why we have the pig and the human in our logo. And translating science to cures is our mantra, and why we're kind of pig-headed and stay with the pig. So to start off first here, type 1 diabetes. This is an example here where you got a little guy, just diagnosed with type 1 diabetes, age 3, bewildered, having great care in this pediatric ward, but you can see there's some distress in him, and it's no doubt. But of course, he says, "But I'm strong, tough, and brave." So, and that's touching to hear. And it's brave children like this that are our number one reason for studying diabetes. So, so we start off with some of what we did. In this case, I'll just show, we had fed omelets and to pigs, and so to speak, and chemically induced diabetes with aloxin, and we did that in Yucatan pigs, and in the Yucatan miniature pig. But what we found is that we could induce diabetes, robust hyperglycemia, but they did not get very obese at all. So, and you can see he's eating his omelet there. And so what does that mean? Are we, I think that we're not kind of falling short of mimicking human diseases, and which would limit the rapid translation of diabetes therapies to the clinic. So what do? Well, you take a step back and take a 30,000-foot view or so, and when we did that, well, we saw an island, and you can see it's a heart-shaped island, and so we just kind of predictive here. So this is maybe a circumflex artery in our heart-shaped island. So it's kind of this perfect symbol for us. So and because on that island were the Ossabaw pigs, and they took a nice boat ride from Spain, from the Spaniards about 500 years ago, and thus had to really survive the feast and famine ecology of Ossabaw Island, and so they had to be obesity prone, and that would then give them a tendency to develop prediabetes and type 2 diabetes. So this may be the quote perfect pig, and this is a harsh environment for them because they're monogastric animals. They can't just eat this nice grass out here to survive. They had so they had to eat a lot to survive the feast and famine ecology, and thus had undergone basically, I think, gene editing for 500 years. And so what we need to really mimic human type 2 or metabolic syndrome and type 2 diabetes is this sort of thing. And here are the changes that go on in humans over a number of years. So, and the diagnosis of type 2 diabetes, we put it at time zero here, year zero, occurs when you have a statistically elevated fasting glucose. But prior to that, there is metabolic syndrome or the insulin resistance syndrome, where what we have is the insulin resistance, and that's hepatic and peripheral, and one has a compensatory beta cell function, increase in beta cell function, such that you can get increased plasma insulin to try and maintain glycemia in the presence of insulin resistance. And so here you see now this impaired glucose tolerance. And then again, once you're diagnosed with type 2 diabetes, you usually have that for the rest of your life. The very important issue here is that comorbidities and complications start right when the insulin resistance syndrome does and metabolic syndrome. So we have really focused at this point for practical reasons with swine models and the amount of time that it takes. And as a matter of fact, to get modest type 2 diabetes, it takes about a year or so of treating those animals. So we want to mimic this in the Ossabaw pig. And indeed, the Ossabaw pig shown here, take-home message is that when they're active and lean, they're relatively healthy, inactive, and have the obesity diet, which, as you might guess, is high in calories, fat, cholesterol, and fructose, typical, quote, Western diet or American diet. So we studied these animals for about 18 years and developed them at Indiana University School of Medicine at Purdue, and then transferred them to CorVus Biomedical, our company. And driving home this issue about obesity. And so here's more about obesity. Here's the poster child for it. And so, but we gave it the best bariatric surgery. We actually, in this case, flew this pig to the University of Washington, and Dave Flum's group studied it with bariatric surgery. And so he got the best care there. So again, to emphasize morbid obesity, here's this is actually a video, and you work with pigs know that as soon as you open their cage, they usually go dashing out. But this pig is just very obese, sedentary, really can't hardly stand up very well, but it really shows the obesity. But she is somewhat modest, but we've also exercise trained these pigs. And so this is about as much as exercise training as I'll give you today. We get a nice training effect with these animals in just 7 weeks. It actually prevented some of their coronary disease. But this is different than a lean Yucatan pig. And you can see here, this is actually more of a track star compared to the Ossabaw miniature pig. Again, he has a similar workload here, time, but they can run up to 75 minutes or so. And now I'm going to go through why the reasons for why we think the Ossabaw pig is the best model, large animal model of metabolic syndrome. And first thing is obesity. And I can tell you, and I'm going to list criteria that are used for humans. And I can tell you that we did a systematic study head-to-head of the Yucatan and Ossabaw. And I'm just citing literature on the Göttingen here in this column. So the Ossabaw and the Göttingen have a much greater propensity to obesity than does the Yucatan. And you can see as I go down the list here with insulin resistance, glucose intolerance, dyslipidemia is shown by increased LDL to HDL ratio. Dyslipidemia is shown by increased triglycerides, and hypertension, that the Ossabaw has a big yeses there in every one of those columns. The Göttingen is pretty good, but the Yucatan really is more of a lean animal, and rather than more for obesity. And finally, what's real important to us, is that do they really develop type 2 diabetes? And it's judged by fasting hyperglycemia. And Ossabaws and Göttingens do, but it is variable for both of those. We don't get really robust effects. And what's important for us also is coronary disease. And the Ossabaw actually has much greater coronary disease than the Göttingen and the Yucatan, at least in our hands. And folks, most of these slides have the PubMed ID down here for you, so you can look these up later. So for a swine model of human metabolic syndrome, I cite this study of Cluzel, which is a HAP endo study, in which they did a systematic review of 6 livestock and 7 miniature swine breeds. And the take-home message was this, and I am going to read this. The Ossabaw miniature swine in particular represents a highly translatable model that develops each of the core parameters of the syndrome, and with many of the associated secondary comorbidities. So, and this was actually, I think, is a pretty strong statement for us because we've never collaborated with the Cluzel group at all, and so it's really more of an independent view of swine models for metabolic syndrome. So yeah, the secondary comorbidities too is a big issue here. So now I'm going to give you some information here about different uses of the Ossabaw miniature pig. And so coronary atherosclerosis first, because that's what we do most. And I want to show this overall experimental design as a kind of a template for doing these studies. And this figures out of our review article in Annual Review of Biomedical Engineering. So and so if you have a lean pig, this is basically the sort of growth curve of that pig over time. And I will say that, let me back up. Is that an advantage here is that these pigs, being miniature pigs, they plateau in their gain of weight over time. So it makes it much better for longitudinal studies. And if you compare then to a domestic pig, though, by 6 months of age, it's 100 kilograms. And so body weight here in kilograms, it's 100 kilograms, and it's still going up. This is not leveling off right away. So it makes domestic pigs difficult to use for that. And I must admit, miniature is a relative term. The Ossabaw pig is miniature compared to the domestic pig, but it's not as small as some people want. Some people want like a teacup pig, and so maybe less than even 10 kilograms or something as an adult. That's not going to happen with the Ossabaw, I can assure you of that. But I think it's a strength and a weakness. The strength being that the Ossabaw and miniature pigs are near human weight, which you really need, in human size, which you really need for some translation to the clinic in a variety of different ways. So do they get coronary atherosclerosis? Well, here's an image of the heart from a lean animal. And indeed, here's from an animal that's been on metabolic syndrome for a diet for about 6 months or so. And you can see the left anterior descending here, and these cotton balls are severe atherosclerosis, almost flow limiting. And we can go through the histology. And so if we look in the Ossabaw miniature swine, just a type 2 fatty streak where you just get a little intimal thickening, completely clinically benign. And it goes all the way up to type 6+ complex lesions with enormous stenosis and calcification, extracellular calcification. And that really does compare nicely to humans, because if you look over here, we get some neo-intimal thickening and more intimal thickening in this sample. And then in this particular instance here, we get very similar calcification as in the pig. So so I want to tell you also about some imaging here too, and this is pretty standard angiographic imaging here, and then here you see actually a focal lesion right there, constriction by the laser pointer. So, and this is necessary to implement more sophisticated imaging that we do, and we've developed catheters for this. And we've done a lot with intravascular photoacoustic and intravascular ultrasound imaging. You do photoacoustic, you also have ultrasound simultaneously. And the strength here is a couple ways. This is circling around, and so you get a cross-sectional view of the artery. And one can do a pullback here, so you can get live imaging of the entire artery and characterize that. And here is a two-dimensional image, and what you're going to see here is we pull back through the artery. In this particular case, there's a fair amount of calcification, which is shown by this acoustic shadowing, which means that the signal drops off peripheral to where the calcification is. And this is fairly serious calcification because it's not likely that this is going to resolve with any therapy. And so when thinking of calcification, I introduce this because there's early calcification. And if you look over here at panel A, this is a perfectly normal vessel. There's just nothing there at all. Few of us are sitting here with all of our arteries that way. But in early, and I'm referring back to this schematic over here. A was the normal vessel. B is where you get some diffuse and intimal thickening, but it's resolvable very well with intravascular ultrasound, and it gives us what we call wall coverage, as shown here. Wall coverage increases over time on some months of atherogenic diet. And then as we work our way up the artery, then in this case, just to make these points, see you have a focal lesion here, and you may have calcification as shown by these areas of acoustic shadowing, and then focal lesion, and then here you see really some more intimal thickening, a substantial amount there. So, but and I'll back up. This calcification right here is not resolvable by statins or anything. As a matter of fact, statins will increase calcification because they clear the artery of other things. And so you want, if possible, to have some earlier indication of the calcification in arteries. And so positron emission tomography combined with computed tomography is a way that we've gone here. And the probe to use is actually a radioactive fluoride, sodium fluoride. So because it binds the hydroxyapatite, which is the major species of extracellular calcification. And if you all brushed your teeth this morning, you probably had some fluoride, and so kind of added some fluoride to your teeth. So, and if you then do the imaging, here you can see calcification, obviously, in the ribs and so forth, but you increase the gain, and then you can study calcification in soft tissues. And so that's what I want to show here. And what we did in a number of studies was combined with computed tomography, so we know where the vessel is, can measure then the F18 signal, and take-home message is that with increasing wall coverage, that is with this just intimal thickening, clinically benign, you get an increase F18 uptake, and so we're resolving really the early steps of calcification even before you see it here in these little von Casa stainings. And so that was really, we've referred to that as molecular calcification, and that intimal thickening. So, and let's get to then the little bit that I'm going to tell you about gene editing. And so here, if you look over here, this is a lean pig, believe it or not. It's an LDL receptor knockout Ossabaw. So on the Ossabaw background, and similar anatomy here, and I guess maybe you can all guess where the left anterior descending artery is here. And this is actually lethal. This is in these pigs when they're homozygous LDL receptor knockouts. Even on a normal lean diet, they will die after a couple years. And so we're actually now breeding to others to get a heterozygote that might not be as severe. And this is compared to over here where we like to see that much atherosclerosis. So, the other thing about the whole genome here is I'm fortunate to have collaborators, Yoli Zhang in China, and Peter Hegard's group in Denmark that did a full genome sequence of the Ossabaws now. So that should give everybody who that sort of tool to make gene editing. So now I want to tell you about coronary molecular targeting as another example of things that can be done in the pig in large animal models. And the way that we did this was doing you can do local gene and drug delivery, and this is called the bullfrog. So it's one of the catheter here. So if I show you up here, the catheter right here, it's got a balloon and it has a microneedle, and so you get it to where you want in the artery, inflate the balloon, and it presses this little microneedle into the wall, so you can inject genes or adenoviruses or whatever you want. I think this actually is cleared for human clinical use also. So we did this in collaboration with Sasha Obokop's group, and to study TRPC channels in metabolic syndrome, because we saw that they were upregulated. And the question is, are they upregulated, or can you modify those and find an effect in the animal? And so here you have an angiogram, so we know where we're putting things here. We put the bullfrog down the artery right here, and here's the needle actually. And so you inject the compound, and in this case, we just did naked cDNA, and so you inject it, and then you have to electroporate it to transfect it into the cells, or transduce it into the cells. And this was an experiment that I thought would fail on two accounts. One, poking a needle in the artery when they already have they're heparinized and so forth. And two, shocking it in the artery. I thought it would just lead to cardiac arrest. It actually was amazing. And so first, what we did was overexpress TRPC5 in the cells. So in lean animals, and overexpressing, because they don't endogenously have much TRPC5 at all. And so this was a really nice positive control. So the lean, and we're measuring calcium then after the fact, after it had been expressed, small signal in the lean, and then in response to histamine, there's a larger signal when you overexpress TRPC5. So a nice positive control. Well, could we knock it down? So what we did was have a metabolic syndrome animal, and then use a dominant negative to one of the TRPCs. And in that case, here we see in response to histamine, an increase larger increase in the metabolic syndrome pig. But then in the pig that we had down-regulated TRPC, it indeed had down-regulated then the calcium response. So can you do gene manipulation in pigs? Well, yes, you can manipulate it at the germline level, but also locally. And I think locally is really the way to go to do some of these things very effectively. All right. Well, I'm going to segue to a different area that is not our main area, but is absolutely fascinating here, and that is bariatric surgery, and in this case, we use cryolipolysis. And I'll bring back our poster child here again, because this animal is very obese, and you can see the central obesity here. This is largely subcutaneous, but this animal does have a lot of visceral fat, which is a major source of the cause of insulin resistance and so forth. So, and it was Rafi Mazer and Alex Babkin that were interested in this and came to our CorVus team to see if we could help them out with this project. They had tested it at their company B2M in a variety of small animal models, and now it's time to go to a large animal like this fellow. So I show you this again because this is another example of how, basically how this study was done. And in this case, what we did at CorVus was for 9 months, we fed them atherogenic diet, so they would get severe metabolic syndrome and look like that pig on the previous slide. We did not do biopsies and so forth, but we did do one intravenous glucose tolerance test right here at the end of the 9-month sort of pre-treatment period. And this was not a prevention trial at all. They didn't use drugs and cells, but they did use their device to do the cryolipolysis. And where am I? Yeah. And so this reversal treatment was not over the entire period here. There was just one treatment with the cryolipolysis, and then followed up with computed tomography scans at 1, 2, 3 times to monitor the steady progression of the disease, the visceral fat increases, or to determine whether it truly was going to be reversed by the treatment. And that was for a 6-month period. So there was a sham group, and I'll emphasize again here, part of this kind of a hybrid study was done at CorVus, and part of it was done then at Indiana University School of Medicine, where we were previously, because the CT scanner was there, and the surgical facility was there at that time. So, and this is the cryolipolysis probe that they've designed, and so here you see visceral fat, and you don't see many of the veins and so forth right here. You can get a little bit of a look at that, but after cryolipolysis, which is just lowering the temperature to 10 degrees C, you can see it kind of turns white there. And the results, I think, were amazing. Here we have at time 0, that is once they were fattened up. Here's the mesenteric fat on the CT scan, and then 6 months later, you can see that it's shrunk. And then here's the sham control, and here where you see the visceral fat really well. And then here's in the cryolipolysis 6 months after follow-up. You can now see those vessels there, and so it had stayed atrophied basically. And some of the really great data are shown here. We did a HOMA-IR, that's a measure of insulin resistance, and so insulin resistance was high in the sham treated ones, but you can see that it was reduced substantially by the cryolipolysis. And then and here's a striking characteristic of the pigs at baseline, basically, is that they have an extreme hypertension, 170 millimeters mercury, and it was amazing that the cryolipolysis also decreased the blood pressure almost to about a normal of 120 systolic over that period. So and now I want to mention aging, and I give you this as a example of a centralized resource. I never thought I would do aging in my life, aging research, because with pigs that live to be about 13 years old, that is not a high yield thing. But because we have a breeding colony, our breeders breed up until they're 8 years old or so, 8 or 9 years old, and so we have animals that are naturally aged. So I'm going to give you just a take-home message from this. We compared young lean pigs, and these are 2.5 years old, so that's a 20-ish year old person, human, and then and we know that if we submit them to a metabolic syndrome diet, they're going to get atherosclerosis. You've seen that before. But in our retired breeders that were almost 9 years old, so 63-ish years old in their 60s, the old lean actually showed an effects here, and mainly with medial hypertrophy. Very little intimal thickening, though, in these pigs. It was not statistically significant. But if you compound it in these older animals by now feeding them the metabolic syndrome diet, the atherosclerosis is phenomenal in them. So they're kind of a synergy of that effect. And so the intimal to medial ratio was extremely high in the old metabolic syndrome pigs compared to the others. And I show this is as a shared resource, and I alert anybody who's interested in it and potentially getting tissue from pigs that we will study in 2024. And yeah, in 2024. So this is a cross-sectional study, and what we have here, we'll just look at all the pigs. We have old pigs, 9 old pigs that average 11.3 years of age. We have middle-aged pigs, thanks to a thing called the pandemic, which may cause the cancellation in the sales of some of an enormous number of pigs that we had in a study. So we ended up with middle-aged pigs that are now 6.4 years. They'll be 6.4 years old starting in 2024, and we'll compare that to young. So we'll do this particular study as a cross-sectional study, and so again, contact us if you are interested in any tissues from that. All right, so insulin pharmacodynamics, and I'm going to kind of increase the pace here. I'm going to come back to our little guy here who, he's strong, tough, and brave, but he's wondering with all that's going on with all these metabolic syndrome pigs and so forth, and he's wondering, says, but what are you doing to help me? And so here's what we're doing. So and let me back up. With well-maintained type 1 diabetic children, especially, they have a propensity to having hypoglycemia. So if you imagine a little kid like this guy who gets his shot in the morning and then just decides, I'm not hungry now, Dad, Mom. And then so what happens then? Of course, he's going to become hypoglycemic. Or if he goes outside, gets really active, doesn't and exercises a lot, that can also induce a hypoglycemia. So it's a pretty serious problem. So we really need some, quote, smart insulin that only acts when only does its thing when glucose is high. So that's we were fortunate that Matt Weber and his team at Notre Dame wanted to do a study with us on their glucose responsive insulin, and so take-home message is that they have an insulin molecule here that has they attached another structure on it, which makes it bind glucose. So have the kind of glucose sensor, and there's this dendrimer diol, and when they are together, insulin is not working. But when glucose increases, it will bind then that site on the when glucose increases, it'll bind that site on the insulin molecule, separates it from them, and then it becomes active. So hence it's going to be glucose responsive. And the results are really quite amazing here. And when we did this study, we did aloxin-induced diabetes, and then did oral glucose tolerance tests in these pigs, and then sampled blood and so forth. And let's start down here with this at we call day -1. So they get have an oral glucose load, and we'll look just first at the glucose. We know they're diabetic because their glucose is about 200 milligrams per deciliter. The glucose increases to about 400 milligrams per deciliter, and it takes several hours to actually return. We treated the pigs with the subcutaneous insulin, smart insulin, and then got these results. And this was after day 5. It was treated. Pigs were treated on day 0, and then even 5 days later from that one subcutaneous insulin injection. Here are the data in response to an oral glucose tolerance test. Blood glucose now is down to about 70 milligrams per deciliter. That is, and that's fasting blood glucose. After the oral glucose tolerance test, it comes up to about 240 or so, but it falls back down to 70 within an hour or so. So highly effective. And the other thing is that there is an insulin response. I failed to mention back here that these are clearly type 1 diabetic because there's almost no insulin response to the oral glucose tolerance test. So really amazing results there. I'm going to give you now a real quick tour because we're running out of time here, but kind of a tour and a how to start a study in swine if you haven't ever done one, or if you want to do one. Here's CorVus, and we have 6 of these different swine units here, and our lab expansion is underway. We have breeding and farrowing unit here, neonatal pigs up to about 6 weeks of age, and this mother has 5 piglets. We only have about 4.2 on average per litter. There's a nursery area where we keep pigs there from 2 to 5 months old. And, we socialize these pigs, start out socializing these pigs right away also. We have a quote grower unit that houses pigs from about 5 to 12 months or even beyond that. And what's very important for studies here is we have several individual housing units that where we can house the pigs individually for control their diet and therapy and instrumentation and so forth. All stainless steel siding here, or excuse me, stainless steel fencing and so forth. So how do you get involved here? Well, if you are interested in these at all, you could do, we have a tissue bank, and you could ask us for tissue to see if your antibodies work, etc. You could quote piggyback. I love that pun, on an existing study and get tissue at the end, fresh tissue at the end of that. Third, you could do a hybrid like Rafi Mazer did with the cryolipolysis, that is, do the metabolic syndrome phase of the study at CorVus and then doing interventions at your site if you wish. And then finally, just let CorVus do the study for you if you need to. So I need to wrap this up and give you conclusions and a main message here. And then here we have my good friend at the bottom. And he is not like this critter here, this mouse. And he's not pretending to be like a mouse and small and that, but he has his advantages and disadvantages. We think one big advantage is he's more like this critter, and this is an obese human. So we think that there's superb mimicry of human metabolic syndrome and comorbidities, and that really allows us to do more translational research, but we can do molecular mechanisms also, and I think we have good shared resources, and then finally, kind of main message is let CorVus do your study for you if that's what you need. So on that, my friend and I say thank you for listening, and you'll have another quiz question now, and then we can take some questions. Thank you.
Truly, truly impactful presentation, Dr. Sturek. Thank you so very much. And yes, we definitely want to launch our second poll. So I'll bring, there we are. We want to find out what type of animal modeling that you're using. And so let's see, we have that poll. Okay, we have that one. I think we did that one earlier, but we'll go ahead and see the results again. Okay, we have 45% that are likely to implement the swine, Ossabaw swine models in their future work. And I think we have another poll that talks about the different types of models that are being used, animals that are being used in your current work. So we'll let Jake get that up and rolling. And while he's doing that, Dr. Sturek, I just want to say I was just impacted by your presentation from a standpoint of what I need to do to up my game in terms of watching what I eat and being able to get on that treadmill. I was quite impressed to know that the Yucatan pig can run up to 75 minutes. That's running, not walking, right?
Running.
Wow, that's quite impressive. Okay, so I did get a message that Jake did launch the other poll. Now we have, what animal models do you currently employ? Looks like that those numbers are pretty high on the rat and swine model. Does this surprise you at all, Dr. Sturek?
Yes. I'm delightfully surprised. Yeah, I think, Patricia, I saw both of the results from both of the questions, both of the polls. So,
Mhm.
so yeah,
91% are using swine right now. What do you want to say to that?
yeah. Well, contact us and use our swine more, I guess.
Yeah. Okay, great. So why don't we go ahead and get started with the Q&A. And I already asked my non-scientific questions about the Yucatan and what needs to be done. But let's go to a question that's been posed. And the question is, what is the major challenge of doing a coronary artery interventional study in Ossabaw miniature pigs?
I'm glad someone asked that because it's not simple to do, and I think the main one of the main problems is that the Ossabaw miniature swine, they're much more sensitive electrophysiologically, than I think other pigs, and by that I mean that we will get arrhythmias and fatal arrhythmias, sometimes when we do a minor occlusion in the pigs. And I think it's very interesting. It's if for someone trying to do ischemia reperfusion, they like for the occlusion to be for minutes, maybe 40 minutes or so. But Ossabaw pigs usually can't handle that, and it's maybe not that they have there's metabolic problems, but that it's the electrophysiological problem, and so they really are more, I should say, more susceptible to sudden cardiac death. And that means a lot more to people when you think of Damar Hamlin, who was sudden cardiac death. Well, these pigs can also experience that also, and so it's just and difficult to bring them back from that. And I think it's a very unstudied area in these pigs that could be pretty fruitful.
Okay, great. Thank you so much. We do have a question from Bill Mueller, and he says, great talk, Mike. He wants to update you on their studies of cannulating the LCX in the IR models. But the question he has is, when will those cross-sectional tissues be available?
Uh, oh yeah, and thanks, Bill, for asking that question. Uh, the cross-sectional tissues from the aged pigs, right? Those will be available we will start those studies in January of 2024, and we'll be sacrificing 2 pigs per week. So there's about 32 pigs, so it'll last about 16 weeks, starting in January 2024.
Okay, and he further
Mhm.
concludes that he can vouch for the sensitivity of the pigs to manipulating the coronaries in IR studies.
Yes, yes. Had some difficulties. So
Okay. And so we do have another question. What is the overall demeanor of the Ossabaws? Since they were feral, I wonder whether they still have wild aggressive tendencies.
that's important also, because it's a large animal. And number one, it's a large animal, so they're a little bit more difficult to deal with. And two, if they're large and they're aggressive, it makes it even worse. So, but what we find is that we do, I briefly mentioned, we do try to socialize these pigs as soon as possible. And we literally pay, a teenager to sit in the cage and basically play with the pigs, and that really makes a big difference. If And so I think they're as docile as any other pig, but if they are individually housed and kind of isolated, they really get kind of they would get kind of an isolation stress, and we avoid
Mhm.
that by having those stainless steel gates that I showed, because the pigs get kind of nose to nose contact, and so they can interact with each other, yet they could still be individually housed. So the short answer there is that you just need to socialize them for them to be reasonably reasonably docile.
Okay, great. We have another question from an anonymous attendee, and there's a word here that I'm not sure how to pronounce, but I'm going to do my best. And it says swine are known to have unique Purkinje cell distribution,
Purkinje, Purkinje.
Purkinje,
Mm-hmm.
Purkinje cell distribution in the ventricles, along with a lack of certain potassium ion channels that make pigs notoriously prone to fatal cardiac arrhythmias. How arrhythmogenic are Ossabaws compared to other breeds like the
Yeah.
Yorkshire, the Yucatan Mini, and the Landers? The
Well,
Landracer?
yeah, landrace. Actually, and that's a great question. That's kind of complements or supplements the question that was addressed, was put forth earlier about doing interventions. Um, the Ossabaws are more difficult. They must have a pretty well developed Purkinje system in their hearts. Um, that could be one reason why it's even more developed in them than other swine, and so we get more arrhythmias in those animals. There's another thing, and my favorite reason for this is that they have an AMP kinase. The Ossabaws have an AMP kinase mutation. It's an impaired function mutation, and we have found that our pigs that have that mutation are much more sensitive to arrhythmias than some other Ossabaws that we have that do not have that mutation. So I think that, and that could be modulating the potassium channels that the questioner referred to.
Okay. Another question from Chong Wen Duan. In all metabolic syndrome pigs, what is the percentage to develop T2D?
Yeah, type 2
And
diabetes.
you want to answer that first, is there's a two
Oh,
part, and what is the blood glucose level in these T2D pigs?
okay. Uh, so yeah, so the percentage that actually develop type 2 diabetes, if we have them on diet, a year to a year and a half, um, they probably uh, probably let's see, um, about 50, maybe 50%. But Chongwen, keep in mind it's not a robust type 2 diabetes. All right, the Ossabaws have a very tight, very tight fasting glucose with very little variability. And so it can be about, say, 62 plus or minus 2 milligrams per deciliter. And so statistically, you can have type 2 diabetes with a statistically elevated fasting glucose of only 75 or something. So it's not robust. That's just what we need to keep in mind there. And yeah, so I hope that answers the question. Did I?
I think you did. And if
Yeah,
not, definitely
yeah.
please feel free to resubmit another question,
Okay.
Chong Wen. We have a question from Eric Mokelke. I hope I pronounced your name correctly. And the question is, do you have any current data on using your LDL-RKO model for creating peripheral artery disease, iliac or femoral arteries, in particular with the LDL-RKO model?
Yeah. I wish we did. Well, we have, because Eric, it is truly it's lethal, that mutation. They'll die after about 2 years, and so we're interested in coronaries. We took that coronary out, examined it. We also did see a massive amount of atherosclerosis in the peripheral vessels. So yes, it could be a really good peripheral artery disease model if it doesn't first die of a coronary event. So it's going to be exciting for us to get heterozygotes to see if we can titrate to that amount of atherosclerosis.
Okay, and Alex Kalk wants to know, what types of tissues do you have currently banked? And he also wants to thank you for such a great presentation.
Oh, well, thank you very much. I'm glad you appreciated it. We have anytime about 50 different tissues. When we harvest tissues from animals, we will collect that many. As soon as I say that, someone asks me for tissue we don't have. So, for example, we stopped collecting some renal tissue. Just contact me and tell me what tissue you want. We will go to our inventory and to look and see if we have it. If we don't have it, we will put you on the list for harvesting that tissue when we next euthanize pigs.
Yes. And I did put your contact information in the chat, both your email and your website, so that people can contact you. And we have one other question from Rafael Ramirez. Are the bullfrog catheters available commercially?
The simple answer, yes.
Okay. All right. And that looks like all the questions that we have for now, and it's very timely. It's 11:57. Do you have any closing remarks that you would like to share, Dr. Sturek?
I do want to do one thing. I'm hoping my Danish colleagues are on. Let's see if I can advance my slide here. Let's see. It's not going. There we go. Because and for my Danish colleagues, to thank them for what they have done, we actually flew pigs to Denmark, because pigs fly, and they established there at the Technical University of Denmark a colony. So our colleagues in the European Union can have a easier access to pigs, and so just contact Peter Hagard at this email address. So we'd be glad to help. And I'd like to thank everybody who attended. And please, if you have any questions, contact us, and we'd be happy to chat with you.
Okay, great. And so what I like to do again is thank you. I like to bring Jake on video, because we couldn't have done this without him as well, Jacob White with APS. And also we put all of your information, Dr. Sturek, in the chat. So if you want to reach Dr. Sturek by email, also you can visit his website, all available in the chat. And so I just want to thank everyone behind the scenes, and of course you, Dr. Sturek, again, for a great presentation, and also for all of you who joined us today. Thanks again.
Thank you all. Thanks, Patricia. Thanks, Jake.